very good deed. My name is SYAFIQA BINTI and I am from Management and Science University. My research title was Preliminary in Vitro Assessment of Probiotic Properties of Lactic acid bacteria isolated from local fermented foods and dairy products. The public consumption of traditional fermented foods and dairy products shows a gradual intensification. This is due to the well-established benefits on human health. The presence of health-friendly bacteria in both fermented and dairy products aids in providing desirable organoleptic properties in improving human health. The linkage between these two factors is due to the presence of probiotic properties. Therefore, the main objective of this research was to evaluate and compare the probiotic potential and safety of lactic acid bacteria strains isolated from local fermented foods and dairy products. As LAB strain shows antimicrobial properties, therefore isolated LAB strains from local fermented foods can be used for future industrial application to suppress the growth of pathogens. Synthetic antibiotics capable to eradicate normal bacterial flora in human body. Therefore, LAB strains aid in minimizing the disruptive effects of antibiotics by reducing the applications and reliance of the synthetic antibiotics. It also helps to inhibit the development of multi-drug resistant of pathogen strains. Therefore, it has become the interest of this study to isolate the lactic acid bacteria from diverse local fermented foods as well as dairy products in order to evaluate their safety and probiotic properties. The methodology of this research is a total of 5 local fermented food and 2 dairy products was collected. The isolation of lactic acid bacteria was conducted on the MRS agar medium suggested by Ramasamy 2012. The phenotypic identification was conducted through gram staining and catalyst tests. For carbohydrate fermentation test, it was conducted as described by Goyal 2012. Moving on to pH tolerance test, the LAB cultures was incubated in the MRS broth with adjusted pH 2, 3, 4, 5 and 6. For the temperature tolerance test, the culture was incubated at different temperatures 4, 25, 37 and 50 degrees Celsius. Meanwhile, for the NaCl tolerance, the LAB cultures was incubated in the MRS broth with adjusted NaCl concentrations 2, 4 and 8%. While diffusion method was applied in antimicrobial activity test, the cell-free supernatant of isolates were tested against gram-positive and gram-negative pathogens. Hemolytic test was conducted as described by Wong 2020. Antibiotic resistance test was conducted using this diffusion method as suggested by Mancani 2014. Lastly, all data collected was analyzed using IBM SPSS for Microsoft Windows. Next, for the results and discussions, from the samples, 11 LAB strains were isolated. However, 6 from them shows gram-positive, catalase negative, non-hemolytic activity, versatile carbohydrate fermentation and shown as bacilli and cocci. These strains were S1, B1, T1, T2, I1 and I3. Under anaerobic condition, LAB are unable to produce catalase enzyme, therefore they do not able to break down the hydrogen peroxide. The capability of isolate to ferment various group of sugars, glucose, maltose, sucrose and fructose indicate that these presumptive probiotic strains are able to survive in different environments. Hemolytic activity of the strains associated with their ability to break down epithelial lining of intestine and considered as the most common virulence characteristic of the microorganism. 6 potential probiotic strains exhibited non-hemolytic activity, thereby validating their safety as potential probiotic strains. Moving on to the antimicrobial activity, all 6 isolates shows equivalent strong inhibitions toward the gram-negative bacteria as compared to the YA, which is the commercial isolates. This antimicrobial property is important for the potential probiotic strains in competitive exclusion of pathogens and maintaining the health microbial balance in GI tract. The organic acids such as lactic and acetic acid, hydrogen peroxide as well as bacteriocins, most of the strains showed higher or comparable antagonistic effects against the test pathogens than the commercial strains. I1 strain exhibited stronger antimicrobial activity against Staphylococcus aureus as compared to YA isolated in this study. In order to be effective in exerting desirable health benefit to human body, it is important for probiotic bacteria to survive in gastroacidic environments. From this test, all 6 isolates show good tolerance from pH 2 to pH 6. Isolates S1, B1, T2 and I1 and I3 were able to tolerate better or comparable to acidic condition at pH 2 and pH 3 with the commercial strains. YA, I1 isolated had significant higher growth in all pH than commercial isolate YA, especially in pH 4. The ability to adapt and survive under environmental stress is vital for the probiotic microorganism. The adaptability of probiotic isolates in lower storage temperature is vital for a better shelf life. From this study, all six isolates exhibited good temperature tolerance from 4 to 37 degrees Celsius. All isolates exhibited significant high growth as compared to commercial isolates in 25 degrees Celsius. I1 and I3, isolated from pickled fish, achieved comparable growth with the commercial isolate at 37 degrees Celsius. Osmotic tolerance is the characteristic that should be displayed by potential probiotic isolates because human gut exhibits 0.9% of salinity. Therefore, it is important for probiotic strains to resist high salt concentration environment to maintain their metabolic activity. From this test, all six isolates showed good NaCl tolerance at 2 to 8% and manifested excellent growth at 2 to 4% NaCl. Three noticeable isolates, S1, I1, and I3 had significant higher growth in NaCl concentration than the commercial isolate YA. For the antibiotic susceptibility test, 100% of isolate shows resistance to amikacin and gentamicin. Meanwhile, 100% of isolate shows susceptibility towards ampicillin as well as the chloramphenicol. All six LAB isolates were resistant to amikacin and gentamicin, implied that these isolates were regarded as intrinsic resistant genes and can be potentially co-administered with these drugs to prevent antibiotic-associated diarrhea. All the six isolates were also sensitive to the broad-spectrum antibiotics, chloramphenicol and ampicillin, which indicated that these isolates lack of any transferable determinatase. These antibiotics are unable to be co-administered with them. As a conclusion, this study revealed that LAB isolates from local fermented and dairy products exhibited desirable probiotic properties and have great potential for future industrial application. However, further identification of isolated LAB using molecular method needs to be carried. For future recommendation, in vivo assessment should be conducted to evaluate